Am I perfectly audible and visible to you all? I believe I'm perfectly audible. Okay, so how are you everybody? Yes, please. How are you everybody? Please tell me. Guys, do let me know. Am I audible and visible to you all? Am I audible and visible to you all? Let me check. Yes, I guess it's a yes, right? Okay, so welcome everybody. Welcome to the session. So it is the part 3 of top 50 concepts of biology, and today we are going to start with the endocrine gland. We will pick up the mechanism of hormone action and the kind of question that can come in that paper. Yes? So am I audible? Am I audible? Okay. So guys, there is a good news for all of you that till March 31st, right, you can avail the Unacademy Plus subscription just at rupees this 4499. Just at rupees 4499, you people can avail the Unacademy Plus subscription. If you know about Plus, then excellent. If you don't know about Plus, so let me tell you what Plus is. But Plus is a platform which will give you the success, that is for sure, right? Here at Plus, let's suppose you know that if you enroll in any of the batch, I hope you know it, and if you don't know it, let me tell you, bache, we have the Nimbus batch for class 11, we have the Nexus batch for class 12, and we have the Phoenix batch for our NEET 2026 aspirants. Okay, this is what we have. Okay, so if you enroll in our batch, first of all, you will have the access of all the batches that are running across Unacademy platform, right? Be it the English batch, Hindi batch, whatever it is, number one. Secondly, you will get the access of the PDF, all the PDFs available, right? Then you will have the access of test series, and at the end, whatever crash course we are going to start, you will get enrolled in that crash course, that too free of cost. So you just need to pay this fees once, right? And for a year, you will be able to avail all the things that will be there on Unacademy Plus platform, right? So that's how this platform works, and trust me, it's amazing. So in the description box there is a link. What you have to do, see this, there is a link, click on that link, and there is some error here, so click on this link and be the part of the batch. Right now there is an error, but right, you can try it after some time. Okay, chaliye. So see this is the coupon code that you people can apply, and you will get the lightning deal. Done, bache. And okay, so let's start the session. And one more thing, right, we still remember our NEET 2025 aspirants. So for you people, NCERT Crux test series is there, bache, and you know that from 27th of March the tests, they are started from 27th of March. So 29th March again part syllabus test, 30th March again part syllabus test, and after that there will be full syllabus test till 29th of April. So do not forget about it and enroll in that particular test series. Okay, chaliye. So let's start the session. So what is the topic that we are going to start first? We are going to start the mechanism of hormone action first. What are we going to start first, beta? We are going to start the mechanism of hormone action first. First of all, let's talk about the endocrine gland. You know that when it comes to our body, right, nervous system is also required and endocrine system is also required. So together, right, they are going to help in the coordination and integration. We know it very well. So when we talk about the endocrine gland, we talk about the hypothalamus, supermaster gland, we talk about the pituitary master gland, pineal gland is also there, thyroid gland, parathyroid gland, thymus is there, my dear students, pancreas, right, which is a heterocrine gland. Okay, so from here only I'll clear your doubts. So as I said, pancreas, what type of gland it is, students? It's a heterocrine gland. What type of gland it is, students? It's a heterocrine gland. So when we are saying heterocrine gland, it is also known as composite gland. What kind of gland it is? it is also known as composite gland. So what is the word that we have? Heterocrine gland, composite gland. It is also known as mixed gland. So what is the meaning of that particular gland? That it is having both. It is having the exocrine part as well as endocrine part. Okay, it is having the exocrine part as well as endocrine part. Okay, both the things are there. And it is not just pancreas. Other than that, gonads are also, the sex organs are also the example of what? They are also the example of composite or heterocrine or mixed type of glands. Okay, then adrenal gland is there, cap of kidney. Ed means adjacent, renal means kidney, so it is adjacent to your kidneys. It is adjacent to your kidney. Right, bache? Right. Yes. Right? Done? Up to this part all clear? Okay, so this is all about the glands that we have. Now, are we going to get the question from this part, types of hormone? Yes, it's a big yes. You can get the question from this particular part as well. Okay, right, that which hormones are peptide hormones, which hormones are steroid hormones, and not just this, there will be a question from the receptors as well. So firstly, let's talk about it, then we will come to this particular part. So basically, you know that whenever you talk about the endocrine gland, whenever you talk about the endocrine gland or endocrine gland, you know that what will they release? They will release hormones. What will they release? Hormones. What these hormones are? They are the chemical messengers. What these hormones are, beta? They are the chemical messengers. I hope you know it very well. So they are basically your endocrine gland. It is also known as ductless gland. Means there is no tube. There is no tube-like structure in which it will release its secretion and then it will pass to the respective part. No, students, no. It is literally not like that. Right? It is literally not like that. Keep it in your mind, please. Okay, keep it in your mind. So it's a ductless gland. It's a ductless gland. So it will directly, like let's suppose there is a gland here. Gland means secretory. It is going to release something. So it is a gland here. It will secrete the hormone. Right? So now through blood, it will pass to its target organ. It will pass to its target organ, right? So they are basically the chemical messengers, right? This gland is going to pass its secretion into the blood. Through blood, it will pass to its respective target. This part clear? This part clear? Now, why is it so, my dear people? Why? Yes. Why is it so? Right? Right. Please tell me quickly, why is it so? Why is it so that it will act on its target organ only? Just say for an example, let's suppose if it is a pituitary gland, which is the master gland, let's suppose it is releasing TSH, that is thyroid stimulating hormone. That is thyroid stimulating hormone. So that thyroid stimulating hormone is going to act on the thyroid gland only. Why is it so? Because of its receptors. Because of its receptors. So receptors are the one that are going to, right, with which that with which that chemical messenger is going to bind. Now, let me give you one more example. You all know about the cell, right? You all know about the cell. So whenever we talk about the cell, we talk about the plasma membrane. Yes, we talk about the plasma membrane. You know that here in the plasma membrane as well, there are proteins which are present. Intrinsic, extrinsic, we know them, right? And these proteins, they play the different role. Like there are proteins, right? There are proteins which will act like what? Which will act like a kind of channels, right? They will act like the kind of channels. Yes or no? Yes or no? They will act like the channels. Through with them, the things will pass. Like let's suppose if this is a water soluble thing, right? It cannot cross through the plasma membrane directly. So here we will have protein channels by which it will be able to pass. Are you getting my point? Are you getting my point? Now here we have the plasma membrane. Okay, so what's going to happen? A hormone, the chemical messenger. Let's suppose it's like that, right? This chemical messenger, but we also call it as like this is the chemical messenger. We can also call it as first messenger. This hormone here is also known as first messenger, right? What are we saying? Yes, bache, it will be the last session. It will be the last session. I told you, see people, I told you this thing specifically, that I will take care of your most important topics. Right now, other than that, not just this top 50 concepts session or series, other than that, I am going to put the short videos for all of you, where you'll be able to revise the chapter in 20 minutes, in 30 minutes. So basically in less than 30 minutes, you will be able to revise the chapter. And in that chapter, I'll be picking up the most important topics, most important MCQs of that particular chapter. So I am there for all of you. And as we said, we are not going to leave you people, right? We want you people to get selected in your exam. But we need your support. We need your consistency, right? I hope you people remember this thing. I hope you know about this particular session as well, right? Just a minute. Let me show you this thing. Let me show you this thing. Just look at this particular session. Complete syllabus in, right, complete syllabus in 15 mega marathons. So again, once again, we are back, right? We are back with our final marathons, right? And you know that, and these marathons are always effective. We are not doing it from last 1 year, 2 years. We are doing it from last many years. And on Unacademy NEET English channel, right, this is for the third time we are going to take such marathons, where in one marathon your complete, right, the complete subject will be completed. So 15 mega marathons are there. So even, let's say, if your preparation is not directional, then this session is for you, right? Then this session is for you. So wait for it. That is tomorrow 8 p.m. you have to watch this session. And other than that, for class 11th, yes, for class 11th to 12th moving candidates, for 10th to 11th moving candidates, I'm coming up with the basics as well. So don't worry, we are here to support you, right? We are not just here for NEET 2025 aspirants. We are here for 2026 as well as 2027 aspirant. When it comes to me, I'm not going to leave you people in between. I'll post your NCERT related sessions, NCERT diagram based session, NCERT data sessions, right? The disorders, the diagrams, everything I am going to cover. And as I said, in less than 30 minutes, right, you'll be able to revise the chapter. I'll put that video. So if you will search that videos, you are going to get such kind of videos on this channel, where tricks are there, where tips are there. So everything is there. And you people can also join my Telegram group, that is Love Biology by Ambika, right? So you can be the part of that. So I'll keep sharing such kind of updates with you all. So now let's come back to the topic, right, bache? So here you people can see that if this hormone, right, this hormone is there in the blood, we also call it as first messenger. So beta, it will bind to its specific receptor, right? Like let's suppose if it is the thyroid stimulating hormone, it will bind to the thyroid gland only, right? It will bind to the thyroid gland only. Are you getting my point? Right, thyroid gland only. Are you getting my point? Yes, please. Are you getting my point? Yes. Done, bache? Done. So this messenger, it will bind to its specific receptor. When I'm saying specific receptor, particular receptor, it's not like that this chemical messenger can bind with any of the receptor. No. When I'm saying specific, a particular receptor to which it will bind. Now, so basically when we are talking about the receptors here, right, are they all going to be on the plasma membrane? No, it's not like that. It's not like that. When you talk about the receptors, there are two. Right, what will be there? There are two types of receptors. Just for an example, the receptors, right, they are cell surface receptors, which are present on cell surface, of course. So cell surface receptors are there. And along with that, my dear people, right, there will be intracellular receptors. Cell surface receptors are there, and then there will be intracellular receptors. What kind of receptors? Intracellular receptors. So cell surface receptor and intracellular receptors will be there. Cell surface, as the name is saying, they are present on the plasma membrane. Here they are present on the cell surface. Intracellular means within the cell. Right? Intracellular means within the cell. Intracellular means within the cell. Right, bache? And to be more precise, they are within the nuclear, or you can simply say nuclear receptors or intranuclear receptors. Okay, these are the receptors that we have to discuss here. So of course there are certain hormones which can cross the cell, which can cross this plasma membrane. So of course their receptors are within the cell, and there are certain hormones which cannot cross this plasma membrane, and their receptors will be there on the cell surface only, right? So we need to differentiate that. This is the first thing. right? This is the first thing. Are you getting my point, people? Are you getting my point? This is the first thing that you need to keep in your mind. Now, what is another thing that we have to take care? Now, just now I used this term, first messenger. What is the term, people, that I have used? I have used the term first messenger, that for the hormone, you people can also use this term. Now, it's like this hormone is not allowed within the cell. Okay, it is not allowed within the cell. So if it has to pass any message, or if it has to make any changes, so firstly it has to bind with its specific receptor. So this is the hormone. What else do we call it? We call it as first messenger. Now of course it has to bring some changes. That's why it is here. So what's going to happen, my dear students? So when this receptor and hormone, they bind within the cell, right, a kind of cascade will start, right? The second messenger will form. So for NEET, the question can come from this part, right? That what are these second messengers? So when I'm saying cell surface, you can call them Soumya, you can even call them plasma membrane receptor. There is no problem at all. Okay, there is no problem at all, right? Clear? So what's the word that we are using here? We are using the word second messenger here. What is the word that we are using here, guys? We are using the word second messenger here. So as I said, this hormone will not be able to cross the plasma membrane, right? This hormone, it will not be able to cross the plasma membrane. So it will bind here with this receptor, and some changes, some cascade of process will be there within the cell, right? It will be there within the cell. Now what's going to happen here? What is going to happen here? So here the second messenger will form. It's just like, let's suppose there is a government office where you people are not allowed, right? You can take this example in this way. They say, let's suppose there is a government office where you are not allowed, right? Because of some security reasons, and you have some kind of information. So what will you do? You'll go there, right? You'll pass your message to somebody, they'll carry it forward. Then accordingly, as per your message, the response will be there. Or let's suppose you make a call, then you tell them, and then accordingly there will be response. Same as the case here, right? Same as the case here, right? Bache, same is the case here. So what's going to happen, my dear people, here? So when this hormone will bind to the receptor within the cell, the second messenger, right? The second messengers will form. So when it comes to the second messenger, your calcium ions, right, your cyclic AMP, CAMP means cyclic AMP, your cyclic GMP, right? But your IP3, what is this IP3? Inositol triphosphate. Inositol triphosphate. What is the full form here? Inositol triphosphate and diacylglycerol, that is DAG. They are the example of second messengers. Do we need to remember it? Yes, it's important. So calcium ion, cyclic AMP, that is adenosine monophosphate, cyclic guanosine monophosphate, IP3 and DAG will be there. Is that clear? Is that clear? So accordingly, the response will be, now let's suppose if this is the hormone and it can cross the plasma membrane, right? Just say for an example, steroidal hormone. Steroids, what are they? They are also the example of derived lipids. Yes, steroids, what are they? They are also the example of derived lipids. So these steroidal hormones, right, they can cross the plasma membrane directly. Because they can cross the plasma membrane directly, they don't have cell surface receptor, right, and they don't have second messengers. Why? Because they can enter within the cell. They don't need any second messenger. They'll directly enter there, they'll directly enter within the nucleus, and accordingly the changes will be there. So here, what we need to remember? We need to remember the examples, right? We need to remember the examples. So here we have some diagrams. So you people can see this. This is the hormone. Let's suppose it is epinephrine. Let's suppose it is epinephrine. It will bind to a particular receptor, right? As per their binding, the changes will be there in the within the cell. Okay. So as per that changes, second messenger will form. Like here, they have given the example of CAMP, that is cyclic AMP, right? They have given the example of CAMP, that is cyclic AMP. Then what is going to happen, people, right? The protein kinase, they will form, inhibition of glycogen synthesis, promotion of glycogen breakdown. So as per it will give the response. Now look at this part, look at this part. Again a water soluble hormone, again it will bind to its cell membrane receptor. Again according to that, within the cell the responses will start. So here again we have the example of your, right, second messenger. So you people can see this. So this is the diagram which is given in NCERT, and yeah, they are water soluble, they cannot cross the plasma membrane. So generation of second messenger, right? So they have just given the example of cyclic AMP or calcium ion, but along with that IP3 DAG is there, right? And CGMP is also there. Fine. Then biochemical responses, then physiological responses, that will include the ovarian growth. That's what you need to keep in your mind. Now the next thing, lipid soluble hormones, they'll cross the plasma membrane directly, right? So you can see that it will enter the nucleus here. There is no requirement for the second messenger, and accordingly the changes will be there, right bache? Right. Yes. Okay, so just have a look. Thyroid hormone, right? So from this example you can just pick up the hint as well, right? You can just pick up the hint as well. You can just have a look, right? So even the thyroid hormones, they can directly enter within the cell, right? So hormone may bind to a receptor before or after dimerization, and that's how it is going to act. And this is the another example of the steroidal hormone, right? So yes, this question can be asked in paper, right? The hormones whose receptor are present within the cell. Steroidal hormone is the example. Your, right, steroidal hormone, your this thyrotropins are the example. So examples are important, right? Examples are important. Just keep it in your mind, people. Okay, just keep it in your mind. Okay, so lipid soluble hormones are also there. So just have a look. Okay, just have a look. Now I'm not going to revise the entire chapter here, but yeah, I'll pick up the most important things. So when you talk about the hypothalamus, you know na, it is also the thermoregulator. We discuss about it in the chapter neural control and coordination. It is the thermoregulator as well. It is the thermoregulator of our body. It controls the body temperature, number one. Plus it is also known as supermaster gland. What do we call it? We call it as supermaster gland. Why? Because it is going to instruct the pituitary. So beta, it will form inhibitory hormone and releasing hormone that will tell pituitary gland to release or to inhibit something, to release or to inhibit something, to release or to inhibit something, right bache? So how can we remember that? First of all, let's talk about the pituitary. This topic can come in your paper. Okay, first of all, let's talk about the pituitary gland. So for the pituitary gland, we also use the word hypophysis. Yes or no? We also use the word hypophysis. Yes or no? Guys, please give the responses in the chat section as well. Yes. Yes, please. Hannah? There can be, right? Hannah? So when you talk about this pituitary gland hypophysis, we discuss it like this. We have adenohypophysis, that is the interior part of the pituitary, and we have neurohypophysis, right students? So when it comes to this adenohypophysis, here we talk about the anterior lobe of pituitary, and we also discuss middle or intermediate lobe of pituitary, right? Middle or intermediate lobe of pituitary. That's what we need to keep in our mind. And we also have here, this one is posterior lobe of pituitary, right? Posterior lobe of pituitary. Okay. So anterior lobe, middle lobe, and the posterior. Anterior one is also known as pars distalis. Middle one is also known as pars intermedia. And this one is also known as pars nervosa. Okay, done. That is what we have. So for this particular thing, anterior lobe, the trick is go to AC parlor. Let's have facial. Go to AC parlor. Let's have facial. What's the trick that we have, people? Go to AC parlor, let's have facial. That is the trick. So when you talk about this trick, G stands for growth hormone, T stands for thyroid stimulating hormone, AC stands for adrenocorticotropic hormone, parlor prolactin. Let's have LH, luteinizing hormone, facial FSH, this follicle stimulating hormone, and we know that we call them as gonadotropins because they act on gonads. So see, it's easy. So this growth hormone is responsible for growth. Thyroid stimulating hormone will stimulate thyroid. This one is will act on another gland, adrenal gland, then prolactin, and this is there. Okay, that is what we have. Now middle lobe will release only MSH, melanocyte stimulating hormone, right? Melanocyte stimulating hormone. So tell me, it's easy now that the middle lobe will just release one hormone, that is melanocyte stimulating hormone. That's it. Now when you talk about the posterior lobe, one question, right? It's a NEET PYQ, my dear people, right? That this neurohypophysis is under the direct neural regulation of hypothalamus. It is the direct neural regulation of hypothalamus. Actually, bache, this is not even considered as, this posterior lobe is not even considered as the true endocrine gland. It is, right, not even, Malika bache, it is not even considered as the what? True endocrine gland. Why? Because it is not making any hormone by itself, right? It is the hypothalamus that will make the hormone with the help of its hypothalamic neurons, will pass it to neurohypophysis. Neurohypophysis will store it and whenever required will release it. Neurohypophysis, posterior lobe. So here we talk about the two hormones. One is your oxytocin, which is also known as vasopressin, right? And the another one is known as oxytocin is not known as vasopressin. It is the ADH that is known as. So oxytocin, love hormone, bonding hormone, then ADH, which is also known as vasopressin. That's the point, right? Having the constrictory effect. So ADH means anti-diuretic hormone. Do we have any doubt here? Yes, please tell me. Right? Oxytocin, right? It is oxytocin, right? Okay, so this is about the pituitary. Now you know the trick. So your hypothalamus, right, will release, I'll give you the example here, releasing hormone. Jaise, let's suppose growth hormone releasing hormone, thyroid stimulating hormone releasing hormone, adrenocorticotropin hormone releasing hormone, etc. Right? Etc. So one more thing that you have to remember, that growth hormone is also known as somatotropin. Right, bache? So hypothalamus will also release inhibitory hormone. So there are three. Jaise dekho, growth hormone is released by pituitary only, responsible for the growth of our body. It is not acting on any other gland. So yes, hypothalamus will have growth hormone, inhibitory hormone, which is known as somatostatin. Other than that, it is also having prolactin inhibiting hormone. Other than that, right, ye dekho prolactin, right, somato, and then melanocyte stimulating hormone, inhibitory hormone. These are the three inhibitory hormones that we discussed. You write it down. If there is any doubt, please tell me. You write it down. Note the flow chart first. Done, bache? Is it done? Chaliye, let's go for that. Let's go for the next topic. So this is about the trick, and here we have the function. So you know that I'll share you this PDF, right? You'll be able to revise it quickly. Just neurohypophysis, right? No hormones are synthesized. Here its hormones are synthesized and stored and released two hormones. Done. Right, done. It is about the posterior lobe. So oxytocin is there, and we have the antidiuretic hormone. Oxytocin is there and antidiuretic hormone. And thyroid gland, yes bache, this topic is also important. Okay, this topic is also important. Okay, so it is the largest endocrine gland. So if somebody will ask you largest gland, then the word will be liver. But when it is the largest endocrine gland, my dear students, then it will be the thyroid gland, right? It will be the thyroid gland. Am I clear? Am I clear? Okay, chaliye. So it is bilobed, vascular it is, right bache. So here you will see it is H-shaped or butterfly shaped, and here these are the two lobes that we have, and in between there will be the isthmus. And this isthmus is non-glandular. It is connecting one lobe with another, but it is non-glandular. It is not at all secretory. That is also a PYQ. That is also PYQ. So overall in thyroid, we have the follicles and the stromal tissue, my dear people. So here you guys can see hypothalamus will release thyrotropin releasing hormone. It will act on anterior lobe of pituitary, which will release thyroid stimulating hormone that will act on the thyroid gland. Okay, and it will release the hormones further. And when you take the cross section of thyroid gland, so here you people can see, right bache, these are the cells, these are that follicular cells, right? This is the colloidal. So usually these hormones beta usually they form, right, in the minute quantity, right bache, they are formed in the minute quantity and then they will be used. But the point is, in the case of thyroid hormones, that is an exception. They can get stored, right, for around 3 months, that is for 90 days, So here you can see the follicular cells, right, and here also we have certain cells. Do you know what is the name of that cells? Right, parafollicular cells are also there. Parafollicular cells are also there that will release your calcitonin. So yes, this is what we have to revise. So tetraiodothyronin, also known as thyroxine T4, triiodothyronin T3 is also there, right? T3 is also there. So they are iodinated, right? Iodinated. Matlab they need the iodine, right? They need the iodine and the amino acid tyrosine. So they stored in the colloid that fills the follicle and are released to the body when needed. Okay, so iodine is very important, right? Iodine is very important. Otherwise beta, they will not be able to form. So follicles, I told you, follicles mane group of cells. So these follicular cells, all of these follicular cuboidal cell, they will release the hormones. These T3 and T4 that will be stored here in the colloids. And there are some parafollicular cells also. Parafollicular cells that will release TCT, thyrocalcitonin, right? So simply you can call it as calcitonin. As the name is suggesting, right, it decreases the blood calcium level. It is having the role in the calcium metabolism. So calcitonin and PTH, parathyroid hormone, ya parathormone, ya Collips hormone, that also release and this calcitonin, they work together to maintain the calcium level in the body. Okay, they maintain the calcium level in the body. That's it. So this is very important. Now beta, we will shift to the anatomy part. Where will we shift? We'll shift to the anatomy part. Let's pick up the topic from this part. And I believe anatomy of flowering plants, it is the most simple. I think it is the simplest topic, anatomy. Right? It is the simplest topic. So when I'm saying anatomy, I'm talking about the study. Study of what? I am talking about the study of internal tissue. I am talking about the study of what? Study of internal tissue. So here, right, you know that the question will come from the sections, right, like root, monocot and dicot root, monocot and dicot stem, and the leaf. This is what we need to understand mainly, right? Meristematic tissue part is very easy, right? Meristematic tissue part is very easy. But what we need to understand, people, we need to understand this part, root, stem, and leaf sections. So first of all, you should know what vascular bundles are. Right, first of all, we need to know what vascular bundles are. So, of course, you are talking about the xylem and the phloem. When it comes to the xylem and phloem, so when it comes to the xylem, it is for water and minerals. It is for water, transport of waters and minerals. So in simple words, we use the word sap here. And the xylem, you know it very well, right? Its direction is like roots, they are absorbing the water, they'll pass it now for the unidirectional. It is unidirectional. Take, and phloem, it is multi or bidirectional. And phloem for food. Phloem for food. What's that, beta? Phloem for food. So it is bidirectional. So it is for the transport of food or the organic substance. So first thing is, when you are talking about xylem, you have tracheids, beta. You have vessels. You have xylem fibers. And you have xylem parenchyma. You have xylem parenchyma. So these three are dead without protoplasm, and this one is living. This one is living. And when you talk about the phloem, so you have what? And remember, the tracheid and vessels. The tracheid and vessel, they are the tertiary elements. What do we call them, people? We call them tertiary elements. They are the one that will help in the transport of water. They are the one that will help in the transport of water. Now when it comes to the phloem, sieve tube cells, companion cells, phloem parenchyma, that you will not see in the case of monocots, and then there will be phloem fibers. So these three are living and this one is dead. These three are living and this one is dead. That's what you need to keep in your mind. Now, let's talk about the arrangement of these vascular bundle. So first is R for radial, and remember, you will see it in the case of root. R for radial, R for different radius or radii. So for an example, when you look at the vascular bundles here, let's suppose this is xylem, this is xylem, this is xylem, this is xylem, right? Let's suppose this xylem. Oh, and let's suppose here you have phloem. When beta this xylem and phloem, they are at different radii, then radial vascular bundle it is. What type of vascular bundle it is? Right, what type of vascular bundle it is? Right, it is the xylem, and the xylem and phloem is present at different radius. Xylem and phloem is present at different radius, right? So R for radial, R for root. R for radial, R for root. This type of vascular bundle you will see in the case of root. That's what you need to keep in your mind. Number one. Take number one. Number two, conjoint vascular bundle. It should be conjoined now. Conjoined collateral vascular bundle. That type of vascular bundle we'll see in the case of stem and leaf. Now here, right, you remember this thing, you will have xylem, right? And phloem, let's suppose. So this xylem and phloem, right? This xylem and phloem, they are at same radius. They are at same radius. They are at what? They are at same radius. They are at what? They are at same radius. Xylem and phloem, like this. Xylem is inside, phloem is towards the outer side. Xylem, phloem, right? That's how they are present. Are you getting my point? That's how they are present. Xylem and phloem. Okay. Okay. So let's suppose if beta, please observe it. Right. Now this type of bundle is the closed vascular bundle. right? It is closed vascular bundle. Why? Because there is no cambium. What this cambium will do, it will help in the secondary growth, like in the dicot stem. It is present. So no cambium, no secondary growth. And here, this thing which is present in between, this is cambium. So when cambium is present, beta, when cambium is present, there will be secondary growth. Am I right? What will be there? There will be secondary growth. The girth will increase. So this type of vascular bundle is open type of vascular bundle. What type of vascular bundle it is? What type of vascular bundle it is? Please do let me know, people. What type of vascular bundle it is? Yes. What type of vascular bundle it is? This one is open. So you can remember it is open for secondary growth. So the vascular bundle which is having the cambium, that will allow the secondary growth to occur. So that is how we can write that it is open for the secondary growth. That it is open for the secondary growth. And this one is closed. So where do you see this? In the case of stem and leaf, right? So this is the very first thing that we need to understand. Baki, other than that, there are other arrangements also, right? So look at this, right? Collateral open, collateral closed. So conjoint, right? Collateral. This is the type of vascular bundle that we have discussed. And bicollateral is also the thing, bache, where when xylem is there in the center, phloem is covering it from both the sides. So it is bicollateral. So it is a characteristic feature of Cucurbitaceae family. ACEAE. Cucurbitaceae family. Okay, that's what you can remember. So vascular bundle thing is important here. Right, vascular bundle thing is important here. Right, now come to the sections. Come to the sections here. Look at this part. So I'm just telling you, if you will get the diagram in the paper, how can you differentiate? So just look at this. This part will be the phloem here. This part is the xylem. This part, xylem, phloem, xylem, phloem, like this, like this. And moreover, because it is root, the arrangement is exact. The xylem arrangement is exact. Xylem arrangement is exact. So exact mein bete, what will happen when it is exact? Right, just say we have two words. One is your protoxylem and another is your metaxylem. One is your protoxylem, another is your metaxylem. It is primary xylem. It is the one that will form first. It is the one that will form later. So protoxylem, right, smaller in size. When that protoxylem, when it faces periphery, when the protoxylem, right, it is towards periphery and, right, so, and metaxylem is towards the center. It is facing that pith, then this type of arrangement is exact. Right, this type of arrangement is exact. Okay, so it's so as per that, see, the young form, the first formed xylem is at the periphery. It is at the periphery. Then from periphery, it is moving towards the center. So this development is centripetal. It is centripetal arrangement. It is centripetal arrangement. Take. And I told you about the endark as well. So endark will be there in the stem, in the leaf, and here your protoxylem faces pith or the center, right? So it is centrifugal. That's it. So in the case of dicot root, if you will see, right, the xylem bundles, this is beta diarch to hexarch, or you can even say tetrarch or hexarch, right? These vascular bundles, they are limited 2 to 4 or 2 to 6. But when you talk about the monocots, right, these are the points which you need to understand. So see, in monocots, there are many. So it's polyarch there. But overall that arrangement is same. Like xylem, phloem, radial vascular bundle, exarch, but here it will be polyarch. Any doubt? Yes, beta Mehran. You can watch my lectures, right? You can watch my lectures, and that will be helpful for you to score good marks. Okay, done. So see, this is polyarc. So if we see the things from the NCRT, the one thing is clear. Outermost layer of the roots, it is epiblemma. It is also known as piliferous layer or piliferous layer. Why? Why? Why is it so? Because here it is without cuticle. It has to absorb the water as well now. So it is what? It is without cuticle. It is what? It is without cuticle. Is that clear? Is that clear? Are you sure? Are you sure? Are you sure? So it is what? It is without cuticle, right? So unicellular root hairs are present that will help in the water absorption, of course, right? Then the cortex is having many layers of thin-walled parenchyma cells with intercellular spaces. And inner layer of cortex is called endodermis, again a PYQ. And here you know that endodermis will have what? They'll have Casparian strips, and that Casparian strips are water impermeable. What kind of strips are they, beta? They are water impermeable. What kind of strips are they? Water impermeable. Is that clear? Is that clear? So it is written here that they have single barrel shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells, they have deposition of water impermeable waxy strips, suberin in the form of Casparian strip. So next to endodermis, you'll have pericycle. So initiation of lateral roots and vascular cambium during the secondary growth take place in the cell. Now, because we don't have the secondary growth in our syllabus, so remember it is the pericycle from where the lateral roots will come out. So that's why here in the case of roots, we say that they are endogenous in origin. Just let's suppose taproot. It is the primary root. They all are lateral root, secondary or tertiary. So how do they originate? From pericycle. So we'll say that they are endogenous in origin. What do we say? That they are endogenous in origin. What do we say? That they are endogenous in origin. Understood? Sure. So pith is small or conspicuous here, right? So in between xylem and phloem, we have the conjunctive tissue. So 2 or 4 xylem and phloem patches can be there. That's it. Or monocot may similar to dicot. Right, epidermis, cortex, endodermis, pericycle, all are there. Okay, so as compared to dicot root, which have fewer xylem bundles, they have more than 6. So do we have any doubt here? Do we have any doubt here? Just focus on the vascular bundle. You'll be able to differentiate dicot root and monocot root. And other than that, my dear students, other than that, you just need, and when it comes to stem and root, then again it's simple, radial or conjoint. So vascular bundle will help you a lot. Okay, the vascular bundle will also help you a lot. Okay, so pith is large and well developed, and they do not undergo the secondary growth. Next. So let's talk about the monocot stem. Now, dicot stem. So when you talk about the dicot stem, secondary growth is there. Right? So basically girth, diameter will increase. So here you have open type of vascular bundle, open conjoined collateral, open conjoined collateral type of vascular bundle, open conjoined collateral type of vascular bundle. And if you will see, right, look at this cortex part here, right? Well differentiated cortex is there. Well differentiated cortex is there. You are not going to see such kind of cortex. People, mark my words, you are not going to see such type of cortex in the case of what? In the case of, you will not see such type of cortex in the case of monocot. It will be randomly distributed. Here you will see pith is conspicuous, right? And that will not be there. Yes, that will not be there in the case of dicot stem. Okay. Okay, so there is a ring of vascular bundle. There is a ring of vascular bundle here. There is, and they are open type. So basically in between these vascular bundle, this part here is the medullary ray. When the secondary growth will start, the cells will become meristematic here, right? The dedifferentiation will occur. The dedifferentiation, you know, na, it is required for the formation of secondary meristem. It is a dedifferentiation which is required for the formation of secondary meristem. So, bache, the speed of the lecture, I hope it's fine for all of you. You know that we have completed it already. So, this is the quick revision, right? So, just remember this thing. So, here the dedifferentiation will occur. What will occur here? The dedifferentiation will occur. Due to that, the cells, right? Due to that, right, here also you will see interfascicular cambium will form. Interfascicular cambium will form. Interfascicular cambium will form here, right? And here, intrafascicular cambium. Intrafascicular, also known as fascicular cambium. So there will be a complete cambial ring here. So that's the pith. That's the pith. Are you getting my point? This is the arrangement that you have. Now, when it comes to the hypodermis, this question can also come, right? Collenchymatous it is. In monocot stem, it will be sclerenchymatous. Then parenchyma, endodermis. So it is a stem. Here in endodermis, there will be no Casparian strips. There will be no Casparian strips, my dear students. Plus here, in the case of this endodermis, because it is having many starch grain, so we will call it as starch sheath. We will call it as starch sheath. Right? We will call it as starch sheath. Then, bacha, endodermis, pericycle. So, pericycle will also be in patches here, phloem, cambium, metaxylem and protoxylem. So, protoxylem is towards the pith. The arrangement here is endark. Right? The arrangement here is endark. So, here you will see pericycle will have that semilunar patches. Okay? Okay? ये देखो. So it is covered with a thin layer of cuticle when you talk about the epidermis, and in the stem we have the trichomes that will prevent the water loss and transpiration, right? And cells arranged in multiple layers between epidermis and pericycle, right? They form the cortex. So the cells which are present in epidermis and pericycle, they form the cortex. So endodermis, do not forget this line, that endodermis is what? Endodermis is the innermost layer of cortex. What's that? It is innermost layer of cortex. Understood? So three subzones are there, जैसे hypodermis, which is having collenchymatous cells just below epidermis, that will provide the mechanical support. Cortical layers which are below epidermis, the hypodermis, sorry, they consist of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. So the innermost layer of cortex is called the endodermis, right? The cells of the endodermis, they are rich in starch grain, and the layer is also referred to as starch sheath. Take? Okay. Okay. So the pericycle present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. Important it is. Right? So read it. If there is any doubt, please tell me. Please tell me. Yes, go for it. Please tell me. Done. Next, monocot stems. The hypodermis is sclerenchymatous here. The hypodermis is sclerenchymatous here. What kind of hypodermis is there? Sclerenchymatous. But vascular bundles are just scattered here, right? You know that there will be no secondary growth. So, closed type of vascular bundles will be there, right? What type of vascular bundles will be there? Closed type of. So, what do we have here? It's simple. Hypodermis is sclerenchymatous. Vascular bundles are scattered. No secondary growth. Closed vascular bundle. Right? Closed vascular bundle. Is that clear? Is that clear? So, smaller vascular bundle will be there at periphery. And rest, the larger one will be there in the center. Okay, they'll be there in the center. So I believe this is something very easy, right? ये देखो dicot stem, very easy it is. This is the monocot stem. Look at the way they are scattered, right? And that is also PYQ. That is also on PYQ. It's not like that I'm randomly touching this topic. No. Look at this smaller one. Smaller one will be at the periphery. So overall the xylem arrangement is going to be in dark. Overall it is going to be conjoined and collateral. It is going to be conjoined and collateral. Conjoined and collateral. But they will be of closed type. They'll be of closed type. Is that clear? Is that clear? So look at this. Look at this. Take, and here we have the comparison. So read the points here. Read the points here. Quick, then we will jump to molecular basis of inheritance part. Do it quickly. Then we'll jump to molecular basis of inheritance part. Do it quickly. Just have a look. If there is any doubt, do let me know. Yes? Is it clear? Done, beta? Okay. Okay. Then this is for the... Okay. Beta, this is for the root. Right? This is for the root. Okay. And this is for the stem. And now when it comes to the leaf, this is the dicot leaf. You know that upper and the lower epidermis is there. Upper epidermis will have, because it is the one that is exposed to sunlight, will have less stomata. Okay, right? Just to prevent that loss of water. Lower epidermis will have more. Okay, xylem and phloem conjoined. Collateral, it is no secondary growth here. So closed type of vascular bundles will be there. So in the case of dicots, you will see that there will be towards upper epidermis side, palisade parenchyma, right, where the cell will be like this, and spongy parenchyma, the cells are going to be like this. That's what we have here. Okay, just so here you have upper and the lower epidermis. Near to the upper epidermis, palisade parenchyma, right, spongy parenchyma, right, the bundle sheath cells. This is the vascular bundle. Then in the monocots, both the sides are kind of same, isobifacial it is. Take, okay, done. So this is all about the leaf, the comparison points that we will discuss, right, that we will discuss when? Tell me, when will we discuss? When are we going to discuss it? When are we going to discuss it? Tell me. We are not going to discuss it. It's simple. I'll share the notes. I'll share the PDF. You'll get it in our Telegram group, Unacademy NEET English Telegram group. But yes, you can join my personal Telegram group as well, Love Biology by Ambika. Chali. So let's go. So before starting this DNA thing, let's quickly revise the nitrogenous bases and everything. So before starting this DNA, what is this DNA and RNA? Nucleic acid, right? We have RNA, we have DNA, right? So so DNA or RNA, what are they? Polynucleotide chains, right? What are they? What do they have? Polynucleotide chain. Poly means many. Now nucleotide. Before knowing about this, we have nitrogenous base, right? We have nitrogenous base. That nitrogenous base will have purines and pyrimidines. So beta purines, 9-membered ring. So one question will come from this part as well. So 9-membered ring, right? Pyrimidines, 6-membered ring. Do you have any doubt here? Do you have any doubt here? So for purines, what is the trick that we have? It's puga. What is the trick that we have for pyrimidines? The trick that we have is cut. So how to remember that? So it's puga. It's puga. G stands for guanine. A stands for adenine. Then nin, mane 9. 9-membered ring it is. 9-membered ring it is. That's how you people can remember. So yeah, guanine is denoted by G, adenine is denoted by A, and when it is the cut, that is CUT. So here we have cytosine, we have uracil, we have thymine. And this thymine is also known as 5-methyl uracil. This thymine is also known as 5-methyl uracil. So in DNA, my dear people, it's G, A, C or T. In RNA, we have G, A, C but U. At the place of thymine, uracil will be there. Important it is. Done. Important is done. So this is for the nitrogenous bases. So 9-membered ring, 1, 3, 7, 9. Nitrogen is present. Nitrogen is present. In the pyrimidine, right? 1 or 3 N is present. Nitrogen is present. ठीक? Now, ribose. So now let's talk about the sugar, which is pentose. Which is pentose. So it is ribose sugar. It is deoxyribose. It is ribose sugar, my dear people. It is deoxyribose. What is it? It is deoxyribose as well. So as I said, they are pentose. They are the monosaccharide. CnH2nOn is the formula. So it will be Cn C5H10O5. But that is ribose. That is ribose. It is C5H10O5. This deoxyribose is C5H10O4. And that is an exception. That is an exception. So let me draw the structure here. So basically it's CH2OH. So which ribose it is? It is beta ribose. Why? Because hydrogen is below, right? At second carbon, you can say oxygen is present, right? So rest it is CH2OH. Rest it is what? It is CH2OH. Take. Now in the case of deoxyribose, how are they different? Right, this is also beta deoxyribose, but here at second carbon, my dear people, right, at second carbon, where? At second carbon, you'll see oxygen is absent. You'll see oxygen is absent. That's how we differentiate. Okay, so in RNA, the sugar that is present is ribose. In DNA, the sugar that is present is deoxyribose. So when nitrogenous base plus sugar they join, you'll get nucleoside. Take for an example, if it is adenine plus ribose, it will be adenosine. So adenosine is a nucleoside. So how can we remember this part? Simple. Nitrogenous base and sugar. Nitrogenous base and sugar. Nitrogenous base and sugar. So adenine, ribose or adenosine. Which bond is there? N glycosidic bond. So same way guanosine, right? your cytidine, thymidine, uridine, they are present. Okay, so if the sugar will be deoxyribose, so deoxyadenosine, deoxycytosine, the things will be like that. Now, nitrogenous base plus sugar plus phosphate, that will be equals to nucleotide. That will be equal to nucleotide. So, in between these two, you will have N-glycosidic bond, and sugar and phosphate will have phosphoester bond. Will have phosphoester bond. So, right, if I have to name it, because one phosphate will get added here. So nucleotide, when it is a single nucleotide, then sugar and phosphate, they'll join with phosphoester bond. Nobody will write phosphodiester here. Phosphodiester will be there in the polynucleotide chain, or when there is the dinucleotide, right? Or when there is the dinucleotide. So it will be adenosine monophosphate. Adenosine monophosphate, or it is also known as adenalic acid. Adenalic acid. Doubt? Yes, doubt. So these basics clear? These basics clear? Are you sure? Done? Okay. Right? Pakka? Okay. So, the basics here, you know that. Look at the structure. This is the pyrimidine ring, which is a 6-membered ring. Which is a 6-membered ring. At 1 and 3, nitrogen is there. 1 and 3. And overall, alternate double and single bonds are there. Alternate या double bonds are there. That's how you can write. So cytosine, uracil and thymine. So thymine is also 5-methyl uracil. It is also 5-methyl uracil. It is also 5-methyl uracil. Is that clear? Is that clear? Baki pyrimidine, a purine ring, 9-membered, 1, 3, 7 and 9. ठीक? So, okay, 1, 3, 7 and 9. Okay, 1, 3, 7 and 9. So guanine and adenine is there. So A for adenine, A for amino. NH2 group is there, beta. At 6 carbon, this is what we have. And in guanine, guanine, O is the thing that you have to keep in your mind. And here you will see C double bond O. NH2 is there at basically 2nd carbon. So that's what you can remember. Okay. And remember when it is the pyrimidine, right? Because it is the nitrogenous base. It will join with the sugar. So it is the N1 that will make the N-glycosidic bond. And when there is the purine, 9th nitrogen will form the bond. That's what you have to remember. I hope the structure is clear. This is the sugar. This is the sugar. And here you guys can see, right? Dekho, nitrogenous base attached to the sugar, N-glycosidic bond. That sugar is attached to the phosphate. This is phosphoester bond, right? But here it is not the monophosphate. You have triphosphate also. So up to this part, it will be nucleoside monophosphate, then di, then tri. Like we have AMP, adenosine monophosphate, diphosphate, triphosphate. That's what we are talking about, okay? Okay, done? Done? Yes, please. Okay, so guys, if you're new to our channel, do not forget to hit the like button. Done? So in the case of polynucleotide chain, like whenever we study anything related to DNA, we say now the synthesis will always be in 5' to 3' direction. So it's simple, right? Let's talk about the see. Sugar is sweet. Everybody is interested in making the bond with sugar. So when you look at this nucleotide, you will see that here in the nucleotide, this is the sugar. Here you have the phosphate and the nitrogenous base. This phosphate is present at its fifth carbon. They say this is the sugar that we have. So you have the phosphate. Let's suppose this. And remember, it is the phosphoric acid, ic. So you can remember it like this. Nucleic, phosphoric, phosphoric. Okay, nucleic, phosphoric. That's what you people can keep in your mind, right? Nucleic, phosphoric. That's what you people can keep in your mind. Is that clear? Sure? Are you sure? Yes? Are you sure? Okay. So this fifth carbon, just CH2OH is written now. So this one will form the bond here, minus H2O, phosphoester bond. So here it will attach. Okay. Here it will attach. Is that clear? Here it will attach. Okay. At fifth carbon. Where? At fifth carbon. Where? At fifth carbon. Now, take. So let's suppose I'll draw the sugar only. I've explained this topic, I think, multiple times. I'm just writing P, not properly the phosphate. Right. Now this is the next sugar, next nucleotide, I would say. Something like this. Take. Oh, sorry, this one is already involved in bond formation. Now, this one will form the bond here. It will be minus H2O. If you will see, this is your first nucleotide. This is your what? This is your first nucleotide. What's that, people? What's that? Yes, everybody. Yes, everybody. It is your first nucleotide. It is your first nucleotide. What do we have here? It is your first nucleotide, right? Now this one is second. So first nucleotide, this fifth carbon will have phosphate. So that is why we write it like this. Now, so whenever we write about the phosphodiester bond, we write it like that. 3-5- phosphodiester bond. Because now you are talking about the polynucleotide chain. So if it is a single nucleotide, my dear people, please pay attention. If it is a single nucleotide, then beta, we are talking about the phosphoester bond. But if it is a dinucleotide chain, right? If there is what? If there is a dinucleotide chain, then there will be a phosphodiester bond. Phosphodiester bond. So here the bond will form 3-5-phosphoester it is. That's how. Phosphodiester it is rather. Take. So let's suppose if it is the 5' end, let's suppose this is the starting of the polynucleotide chain. This is the end. So 3' end is also having the OH. 3' end is also having the OH. 3' end is also having the OH. So 5' end will have the phosphate, 3' end will have the OH, and the synthesis will always be in 5' to 3' end. Right? And in the case of DNA, you know that the one strand is 5' to 3', another will be 3' to 5', ATCG. This is normal. Adenine will form 2 hydrogen bonds with thymine. Cytosine will form 3 hydrogen bond with guanine. So basically, we talk about the Chargaff's rule here. We talk about the Chargaff's rule here, right? We talk about the Chargaff's rule. Look at this picture. Whatever doubts you have, just post it here, right? Just post it here, quick. Just post it here, everybody. Done? But position of carbon, what? This is the third carbon, 3 dash, 5 dash. This is the fifth carbon of next sugar. Right? This is the third carbon of first sugar. This is the fifth carbon of next sugar. So that's why it is 3 dash 5 dash phosphodiester bond. Right? 3 dash 5 dash phosphodiester bond it is. Hanji, doubt? Yes. Any doubt? Any doubt? Sure? You don't have any? So this is one more diagram that you have in front of you. Just see this. If there is any doubt, please tell me, people. Please tell me and be quick. Yes, please. Doubt? Okay. Okay. ठीक चलिए let's go. So this is the structure of DNA. So yes, they do ask the question from this part as well. Right? The DNA is a long polymer of deoxyribonucleotide. So how can we check the length of DNA? Number of nucleotides. Right? It is checked as number of nucleotide right present in it. Total number of nucleotides will tell us about the right, Piche? Total number of nucleotide will tell us about the length of the DNA. Now, a bacteriophage psi into 174 has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs, and E. coli has 4.6 into 10 raised to power 6 base pair, and haploid content of human is having 3.3 into 10 raised to power 9 base pair. Okay? So if diploid content, they'll ask 6.6 into 10 raised to power 9 base pair. That is also PYQ. So human DNA, the length is around 2.25 meter long. 2.25 meter long basically. But my voice is not clear? Is it so? Voice is not clear, is it so? Is it so? It's clear, right? Okay, done. So this part is clear. This part is clear. Okay. So the bonding part we have discussed. This picture. So 5 dash end. Let's suppose if the question is image based. So 5 dash end will have phosphate. If at the place of that they have given the OH, that will be incorrect, right? So 3 dash end will have hydroxyl. 3 dash end will have hydroxyl. So look at this long chain here. This is important. This is important. This diagram is important. Next. Okay. Baki beta, these discoveries are also genuine, but like you can make the short notes, and I'll take a session where we will discuss about what? We will discuss about the, right there, we will take a session where we will be talking about the scientists only. All the scientists that are given in NCERT, we'll discuss that. So DNA, is that an acidic substance? Yes, ma'am. Present in the nucleus, it was first identified by Frederick Mischer in 1869. It was identified by Frederick Mischer in 1869. Okay, is this information important? Yes. He called it as nuclein because he saw it in the pus cell, right? Because he just saw it in the pus cell, that is in the WBC. Okay, so due to its acidic properties, right? Due to its acidic property, later on, Altman, right? Later on, Altman named it as nucleic acid. Altman named it as nucleic acid. Okay, bache. So how do we get to know about the structure of this particular structure? Right, the main contribution of the, see, no doubt, in 1953, right, Watson and Crick, they got the Nobel Prize for that, right? So basically, no, it was in 1953, Watson and Crick, based on X-ray diffraction data produced by Morris Wilkin and Rosalind Franklin, right? They proposed this thing, and later on they got the Nobel Prize for that. So let me tell you, if I have to discuss about the contribution mainly, no doubt we talk about the Morris Wilkin and Rosalind Franklin, right? And X-ray diffraction is also known as X-ray crystallography. They're photo 51. Right? So actually the main lady behind is Rosalind Franklin. Do read about her, right? It was mainly her work, right? She didn't get the recognition. So Rosalind Franklin, she was the one who took that picture, photo 51 it was, right? She was the expert of this X-ray diffraction, right? She was, the Morris Wilkin was working with her, right? He, without her knowledge, passed the data to Francis Crick, so to James Watson and Francis Crick, and they just explained that bonding part, right? That base pairing part, the bonding part, and then they got the Nobel Prize for that. But she is the main lady behind. She is the main lady behind. It's just that she didn't get the recognition. Okay. Okay. So one of the hallmarks of their proposition was base pairing, and it was Irvin Chargaff who proposed that. So you know that the amount of purine is equals to pyrimidine, and mainly adenine is equals to thymine, cytosine is equals to guanine. That's what we have to keep in our mind. Okay. So the ratios between adenine and thymine and guanine and cytosine are constant and equals to 1. Okay. Chaliye. So this part clear, bonding part clear. So here you can see the diameter here is 20 nanometer or 20 angstrom, right? You know that when each base is bonded to another, we will call it as base pair, right? And in between them, one base pair will stack, the another one will stack over it. So the distance is 0.34 nanometer, which is also known as 3.4 angstrom. So basically, the information that I'm discussing, it is of BDNA, right? Which was the most commonly occurring DNA, also known as right-handed DNA. So this is the one that Watson and Crick has explained. And your Z-DNA is actually beta zigzag DNA. What kind of DNA it is? It is actually the zigzag DNA. Am I clear here? It is actually the zigzag DNA. Am I clear here? Yes? Sure? Sure? Are you sure? Any doubt? Yes, please. Any doubt? Yes? Done, beta? Any doubt? Okay. So this is about the bonding. Done. Baki, this is the central dogma of information. Who gave it? Francis Crick. Right. So it is NTA's favorite question. Trust me, it is NTA's favorite question. They simply love this. So DNA, it replicates. DNA will form mRNA, that is transcription. mRNA will form protein, that is translation. So this is the central dogma of information. Now when you talk about the transcription, in the case of prokaryotes, beta, the site is, right? The site is what? It is cytoplasm. The site is what? It is cytoplasm, right, bache? So it, in the case of eukaryotes, what is the site? That will be the nucleus. What will the site? Nucleus. Translation, in both the case, the site is cytoplasm. The site is cytoplasm. Are we clear here? The site is cytoplasm. Okay. Chali. Next part. In some viruses, the flow of information is in the reverse direction. So beta, we call that as central dogma reverse. What do we call it? We call it as central dogma reverse. The another word for that is feminism, and it is basically the reverse transcriptase, right? That will do the reverse. It is the reverse transcriptase that will do the reverse transcription here. Is that clear? Is that clear? Sure. So just have a look of this DNA and RNA comparison. Then let's talk about the packing, right? DNA packing. Quick. Let's talk about it then. Hello? Answer please. What kind of rattling noise? Is that okay? Fine? Yasir, aapko bhi Eid Mubarak. Done? So DNA, RNA, just look at the comparison. Any doubt? Yes, please. Any doubt? The sound should not be there. Even I don't know why is it so. Okay. Chaliye, let's move to the next part. Okay. Now this is how the arrangement is. Your DNA will be coiled like this, right? And I believe you remember that when you talk about this type of structure, you will only see your chromosome as compact one. You know, na, this is your chromosome. This is centromere, also known as primary constriction. This is what? This is the sister chromatid. What's that? It is the sister chromatid. So centromere is there. Sister chromatid is there, right, bache? So we will see such type of chromosome, right, when the cell is in the dividing phase, right? You know that it is the prophase in which that compaction will occur, right? Overall, when you talk about this DNA, it's a 2.2 meter, right? It's a 2.2 meter long polymer, and we have to fit it in a nucleus where this is the size of the nucleus. So we have to pack it. So first of all, we need to know about the histone proteins. I'll come to the topic directly. What kind of proteins are they? Basic, means they are positively charged. What kind of proteins are they? They are basic. They are positively charged. Now here we have the basic amino acids, right? And here you will have lysine and arginine. Right. So overall, when it comes to the basic amino acid, you have histidine, arginine, lysine. You have histidine, arginine, lysine. You have histidine, arginine, lysine. But here you are talking about the histone proteins, and it is lysine and arginine. Okay, so you will have, see, H2A, right? I'm just making it randomly this. So basically, what will you have? Two subunits of H2A, two subunits of H2B, two subunits of H3, and two subunits of H4, right? What do we have here? H2A, 2H2A, 2H2B, 2H3, 2H4. They'll form octamer, money 8 subunits total, right? So 2 copies of H2A, 2 of H2B, 2 will be of H3, 2 will be for H4. Now, your DNA will coil over it, right? Your DNA will coil over it. Now we are calling it as nucleosome, right? When DNA is coiled over it. Initially, it was just the octamer, right? When DNA will coil over it, right, then we are calling it as the nucleosome, right? Now there will be, so just look at it, right? Your DNA, which was 2 nanometer, that is 20 angstrom, it will become 11 nanometer. So this is the chromatin. Your DNA is coiling over the histone proteins. So it looks like the beads on a string. And this is your first nucleosome. This is your another. In between, beta, we have the linker DNA, and this one is transcriptionally active. So here, with this linker DNA, right, we have our H1 associated, that is the linker histone protein. That is the linker histone protein. Which protein it is? It is the linker histone protein, right? H1. So it's not the part of octamer, right? Right, bache. So that is how it is. So it will become 11 nanometer. This is your chromatin. Then it will further coil, right? Then you will call it as solenoid model. Solenoid model. It will become around 30 nanometer. So nucleosome will fold according, right, into a zigzag fiber solenoid that will be having 30 nanometer in diameter. Then beta, radial loops will form that will be having 300 nanometer. Then they will form such type of compact structure. So when it is like this, 700 nanometer. When it is like this, 1400 nanometer. So imagine we started from 2 nanometer, we reached here. So this condensation will occur. Okay, so initially these histone proteins are there. Later some non-chromosomal protein, right, will be there. NHC, right? Non-histone proteins. They are, initially they are histone, but later on some other proteins will be there for the further packaging, and they will be acidic. They will be acidic. Okay. Okay. So it is also an important concept. Done? Done, bache? Any doubt? Any doubt? Sure? Okay, so let's go. So again, this is the diagram. Please have a look. Please have a look. Quick. Anything else? Yes, but in the prokaryotes, right, you will have some other basic proteins that are polyamines. You will not talk about these proteins that we have just now discussed. Okay, you will have polyamines. They are also basic. They are also positively charged. But here you are not going to talk about these histone proteins. Okay, done. NHC, non-histone chromosomal protein, that will help in further packaging. So HSP sir is there in the session. Welcome HSP sir. How are you? Hello HSP sir, how are you? Long time no see. We miss you. We genuinely miss you here. Yes, please. Any doubt from the diagram? Right? So 2 nanometer to 1400 nanometer. Is that clear? Okay, so next is when you talk about the length of the DNA, it is equals to the number of nucleotides. It is equals to the number of nucleotides. So, the distance between two consecutive base pair is, remember, 0.34 nanometer. So, if I have to convert it into the meter, so it will be 0.34 into 10 raised to power minus 9 meter. So if we have to check the length of DNA, is it important? Yes, it is important. So in your short notes, kindly mark it. What we have to write, it's simple. Total number of base pair. Let's say total number of base pair. Base pairs they are saying. They are not saying bases. Total number of base pair with distance between two consecutive base pair. Right? So 6.6 into 10 raised to power 9 base pair. Total number of base pairs that we have in the diploid DNA. Thank you so much, sir. HSP sir. Long time no see, sir ji. जल्दी मिलेंगे फिर से. So we have to multiply it with 0.34 into 10 raised to power minus 9 meter. Okay, why do we multiply it here? Because, right, no doubt we know the total number of base pair. We have to consider the distance between two bases or base pair. Then only we'll be able to get the length of the DNA. Okay, we will be able to get the length of the DNA. So that is what you need to keep in your mind, guys. Take, so this diagram important. Now we'll directly move to the Griffith's transformation experiment. Now I'll ask you a few questions. When we talk about this transformation experiment, so from this experiment, do we get an idea that it is the DNA which is the genetic material? Yes, people. When we talk about this transformation experiment, which is also known as Griffith's experiment, transformation experiment, which is also known as Griffith's experiment. So do we get the idea from this experiment that it is the DNA which is the genetic material? Yes, that it is the DNA that is the genetic material. Yes, please. Yes, that it is the DNA which is the genetic material. Do we get that idea? Yes or no? Do we get that idea? Yes? Nobody is answering. Chalo, anyhow, your wish. So, no, we will not get this idea. When we are saying transformation, let's suppose this is a DNA. This is a cell. Let's suppose if this cell is picking up the DNA from the surrounding, of course, that DNA will become the part of its own DNA, right? When it will accept it from the surrounding. And when it will become the part of its own DNA, it will make the protein that will give the unique property to this particular cell as well. This is the transformation. When the cell will pick up the DNA from the surrounding, when it will incorporate that DNA into it, right? When it will incorporate that DNA into it, right? Then it will show the transformation. Now, when you talk about the Griffith's experiment, we talk about the bacteria here. Which bacteria do we have here? Streptococcus bacteria. So, coccus means spherical. Coccus means spherical shaped bacteria. Spherical shaped bacteria it is. It is also the pneumococcus. It is the one that causes pneumonia. But here, students, we have two strains. We have S strain, that is our smooth strain, right? Why it is smooth? Because it is having the capsule, polysaccharide capsule that causes the pneumonia. And it is the R strain as well, rough strain, right? It doesn't form smooth colonies. It doesn't have that polysaccharide coat that will cause the pneumonia. So this is what we have. This is what we have. This is what we have. So this rough strain, it is non-virulent. It will not cause pneumonia. That will not cause the death of this mouse, right? It doesn't cause pneumonia, right? Because it doesn't have that polysaccharide coat that can cause pneumonia. So when we inject it into the mouse, mouse will live. Smooth strain, which is virulent. Virulent means which is pathogenic. Which is pathogenic. Now what's going to happen? It will kill this mouse. When it is heat killed smooth strain, matlab when you heat that S strain bacteria, the capsule will get destroyed. When you will inject that into the mouse will live. But another experiment, this fourth experiment was the one, right, that shocked Mr. Griffith. That rough strain, it is also non-virulent. Heat killed S strain, it is also non-virulent. Then when he injected the combination of both in the mouse, it dies. Why? Because both are what? Both are non-pathogenic. So he came to a conclusion that there is something that has transformed. There was something in heat-killed S strain that has transformed the rough strain into the pathogenic one. Because later on, this rough strain, when it was figured out, many experiments were conducted, then this rough strain was making what? It was making the colonies. It was synthesizing that capsule, right? So from this experiment, no doubt, we will not get the idea that it is the DNA, right? We will not get the idea that it is the DNA which is the genetic material. But yeah, it was clear that there was something in that heat killed S strain that has transformed the R strain, and of course that is quite stable because heat was not able to destroy it. So yes, the experiment year in 1928, Frederick Griffith. Do we need to remember it? Yes, it is important as well, right? So what conclusion they had? That the R strain bacteria had somehow been transformed by the heat killed S strain bacteria, right? So some transforming principle transferred from S strain, heat killed S strain, that enabled the R strain to synthesize the smooth polysaccharide coat and made it virulent. Okay. So after that, we talk about the biochemical characterization of the transforming principle. After that, what do we discuss? We talk about the biochemical characterization of the transforming principle. What do we discuss, people? Biochemical characterization of the transforming principle. So who gave that? Oswald Avery, Colin MacLeod, Macklin McCarthy, right? Macklin McCarthy. So the genetic material was thought to be a protein. They worked to determine its biochemical nature. So they purified what? All the protein, DNA, RNA from that heat killed S cell. They checked it and they tried to check that which transformed that R strain into the S strain, right beta? So they discovered the enzymes. So let's suppose first of all they isolated everything from that heat killed S strain bacteria, then they tried to edit the enzymes. So let's say for an example this. Here DNA, RNA, protein, everything is there. So if I will add RNAs here, the enzyme that digests RNA. So of course the transformation will occur. If I will add protease here, the transformation will occur. But if I will add DNAs here, the transformation will not be there because it is the DNAs, my dear people, right? Why is it so? Because it is the DNAs. It is the DNAs that will eat or the digest DNA, right? That will eat or the digest DNA. So ye dekho. So they discovered the protein digesting enzymes, proteases, RNA digesting enzyme, RNases. So these two will not affect the transformation, but digestion with DNAs will inhibit the transformation, suggesting that DNA can cause the transformation. Okay, but many scientists, they were not agreed to that. So no issues. So here you guys can see they treat with, so it is having what? So S cell spun to bottom of the tube, kill with heat, homogenized cell, recover S filtrate. So when they treated it with deoxyribonuclease, right? So no transformation was there. When they treated it with ribonuclease, transformation was there. When they treated it with protease, transformation. So that's what you have to keep in your mind. So that unequivocal proof that it is the DNA which is the genetic material, it came from the experiment of Hershey and Alfred Hershey and Martha Chase, 1952. So they experimented with E. coli and bacteriophages. So what are bacteriophages? Viruses that infect bacteria, and this is a double-stranded DNA virus. So now we need to talk about this experiment. So do you have any doubt here? Right? Do you have any doubt? Yes, please. Do you have any doubt? Any further doubt? Do you have any doubt? Sure? Okay. So, in this experiment, what they did. So, first of all, right, I'll tell you about the setup. Just let's suppose if this is the bacteria. I told you it is E. coli, Escherichia coli, right? They were experimenting. They were infecting this bacteria with this virus, double-stranded DNA virus, right? Right. Now, no doubt, we know now it is the DNA which is the genetic material. We are just reading about their experiment. So, you know that DNA is having phosphorus and you know that the protein is having sulfur, right? You remember? CMS. Do you remember CMS? Cysteine, methionine. They are sulfur containing amino acid. Cysteine, methionine, they are sulfur containing amino acids, right? Sulfur containing amino acids they are. Now, so they tried to grow, right? What they tried? Like they had that bacteria. So they conducted this experiment, right? In basically they conducted two different experiments. In one experiment, they used the radioactive sulfur, right? Radioactive sulfur S35. In another experiment, they used what? Radioactive phosphorus P32. Okay, so phosphorus will inculcate in DNA. This one will be in the protein. And here, that is what we have. This is the structure of that virus. It is obvious, beta, right, that it is the DNA which will pass to the E. coli, which will pass to the next generation. So let's have a look. Let's have a look, right? Just look at it. So you can see protein codes, they are radio labeled, right? Because firstly, we have to grow this virus in a medium where radio labeled S35 is there. Now when this experiment will be conducted in 3 steps. The first will be infection. The second step will be blending. The third step will be centrifugation. First step we'll try to infect that bacteria with virus. In second step, we'll blend it, right? Because we want to break their contact, right? And then in the centrifugation, we'll separate their components, right? So in the first step, they will be infected. Of course, radioactivity will not enter in the cell. Why? Because we know that it's not in the protein, but it is in the DNA. Then how will they get to know about it? They'll do the infection, then blending, then centrifugation. After the centrifugation, right, what will they do? You know that this one will be something at the palate, at the base, and this upper portion will be, lighter one will be supernatant. Now, because it is the lighter things will come at the base, right? The lighter thing in the centrifugation, the lighter thing will come at the base. So you know that the size of the virus is quite smaller than the bacteria. So viral component will be, right? The bacterial component will be there at the base and the viral component will be there in the supernatant. So when they use the radioactive sulfur, the radioactivity was there in the supernatant. And when they use the radioactive, right, phosphorus, right, because it is a DNA that will pass to the E. coli. So of course, it is the part of radioactivity was there in the pellet. It was the part of bacterial component actually. Okay, it was the part of bacterial component actually. So that is the infection. That is the process. So bacteriophage will infect E. coli growing in a medium P32, which labels DNA as red, matlab radioactive. Now when these bacteriophage, they will replicate within E. coli, they will form new bacteriophages. Now they will infect the unlabeled bacteria. After that, blender will be there, right? Agitation will be there. Placing the newly infected bacteria in a blender and agitating will remove that bacteriophage's body, right? The genetic information, including the labeled DNA, will be there in the cell. So blending may we have to break the contact. We have to break the contact in between the bacteria and the bacteriophage. What we have to do? We have to break the contact in between bacteria and bacteriophage. Then after that, what is going to happen, my dear people? After that, what is going to happen? The lysis will be there. So phages will reproduce. The progeny will have the labeled DNA. From parent lysis releases partially labeled progeny, right? Then we have to infect the other cells. So here what they did the same, but here the protein was labeled. Protein was labeled. Look at the 3 steps here. So radioactive sulfur, radioactive phosphorus, it will be P32. So right, focus on the right, please focus on the radioactive word here. So S35 P32, you should know the isotope as well. So radioactivity was there when it was S35, it was there in the supernatant. So can we get this type of question? Definitely it's a yes, right? It is a yes. You will get such kind of question in the paper. Okay, so no radioactivity was detected in the supernatant. No radioactivity was detected in the supernatant. Understood? Understood? So next is beta Michelson and Stahel's experiment to prove the semi-conservative nature of DNA replication. So the kind of question can come. So he used 15NH4Cl. So it is the heavy isotope of nitrogen, not radioactive, right? So he grew the E. coli in a medium having this 15NH4Cl for many generation, right? When both the DNA become 15N, then what he did, he changed the medium, he passed it to the 14N. So first generation. Why it is after 20 minutes? Because the generation time of E. coli is 20 minutes. Bidirectional DNA replication is there. Bidirectional DNA replication is there. And due to that, yes, people, due to that bidirectional DNA replication, yes, due to that bidirectional DNA replication, right? In 20 minutes, E. coli will divide into 2. So we got the hybrid. And after 40 minutes, this is what we got. So the density gradient cesium. So with the help of the density gradient cesium chloride centrifugation method, right, they got the bands and accordingly they proved. Other than that, if somebody will ask you who gave the experimental proof for the semi-conservative nature of DNA replication in eukaryotes, then it will be Tyler and his colleagues. Remember? They used the radioactive thymidine, right? They used what? They used the radioactive thymidine, okay? They used what? They used the radioactive thymidine. So do we have any doubt here? Do we have any doubt here? Yes, Tyler and his colleagues. Okay, so next topic that we need to understand is the DNA replication, okay? But before the DNA replication, let's have a quick revision of one more important topic. Let's talk about Mr. Mendel, father of genetics. So, you know na, 7 characters he selected. So if I'm saying 7 characters, the alternative form of character is trait. So 14 trait in totality, 7 contrasting character, or you can say that 14 traits, right? So yes, we should know this thing, that stem height, flower color, flower position, pod shape, pod color, seed shape and seed color. These are the 7 characters that he selected. That so stem height may, you know that the dominant one and the recessive one, and their chromosome number is also there. So basically these characters, they were present on chromosome, different chromosome. So he didn't get any post-Mendelian inheritance issue there, right? Right? Okay. Okay, so yes, any doubt? Yes, please, any doubt? If there is, please tell me. हाँ जी. Sure? Are you sure? Okay, let's go there. So, what do you understand with this particular topic of DNA replication? What do you understand with this particular topic of DNA replication? So, do you need break or is it fine? I think it's fine. Hannah, it's fine. Without break, I think it's fine. I hope you all are done with your dinner. Okay, so let's talk about the DNA replication, and we know that it will take place in S phase of interphase. S phase of interphase, right? We know that DNA content will get doubled. DNA content will get doubled, but number of chromosome will remain same. But number of chromosome will remain same. Right? Right? So let's talk about this DNA replication. And number 3, it is semi-conservative in nature. And of course, we need a lot of enzymes for that. Okay. So in our syllabus, that the DNA replication that we study, it is of E. coli, Escherichia coli, right? Matlab, we talk about the prokaryotic DNA replication in detail, right? So there are a few differences when we talk about it. So you know that if it is the bacteria, that circular DNA is there. Circular DNA is there. So it's not like that ki replication can start from any point. In a very simple language, I will be discussing this topic. Trust me on that. Right? In a very, very simple language, I am going to discuss this topic. So kindly pay attention. So you have, what type of DNA do you have? You have circular DNA. What type of DNA do you have, people? You have circular DNA. What type of DNA, guys, do you have? Yes, please. What type of DNA do you have? You have circular DNA, right? You have circular DNA, okay? Right? So in this circular DNA, there will be only one side, right? First of all, it is the DNA, it is the circular DNA. We have to copy the information given in this DNA, because you know that there is a semi-conservative mode. This DNA is having all the information. So we need to open the DNA first. So the replication will not start from any point, right? It will not start from any point. So basically you have to understand that the replication will always start from origin of replication, right? There is an origin of replication from where the replication will start. So it is ORIC site in the case of E. coli. In the case of E. coli. So it is the point from where the replication starts. From where the replication starts. From where the replication starts. It's like that, right? It's like that. Now, in prokaryotes, when you talk about what? When you talk about what? When you talk about the prokaryotes, in prokaryotes, there is only one origin of replication. So, or a replication. So we call it as monoreplicate. Why? Because there is one origin of replication. Why? Because there is one origin of replication. Why? Because there is one origin of replication. But in eukaryotes, because the DNA is linear, so they are poly or multi-replicant. Poly or multi-replicant. Many origin of replication. Many origin of replication. So firstly, there should be a origin of replication. Now, we'll discuss about the enzyme helicase, which is also known as amylase. This is the enzyme that need the energy from ATP hydrolysis, right? In a very short and simple way, we will discuss this topic, and it needs magnesium ion. Magnesium as a cofactor. So what is going to happen here, my dear people, right? What is going to happen? What is going to happen? Please tell me. Please tell me. Yes, please. What is going to happen here? So no doubt there is an origin of replication. The helicase has to come. It has to open the DNA because we have to copy the information given in it. So helicase will come, which is also known as unbindase. It needs the energy from the ATP hydrolysis, so it will open the DNA. But remember, the DNA will not open in one go. So let's suppose, imagine, do I have bag? No, I don't have my bag right now. So just say, let's imagine there is a bag. If you're trying to open it, like let's suppose you're trying to unzip it, you're trying to open it, you'll do it in one go. But here it is not going to happen. It is not going to happen because it is a very, yes, this process, because this process requires high amount of energy, right? The high amount of energy is required here. Very high amount of energy is required here. Are you getting my point? Yes, please. Are you getting my point? Yes, please. So because there is an energy demand here, so that DNA will open slowly, slowly. That DNA will open slowly, slowly. Right, bache? Just for an example, this is what we make Y-shaped replication fork. So this is 3 dash to 5 dash. This is 5 dash to 3 dash. So you are trying to open the DNA. You'll open it slowly, slowly. Slowly, slowly these hydrogen bonds will break. So that is why, what do we get here? We get this Y-shaped replication fork here. We get this Y-shaped replication fork here. Now, because we were talking about the prokaryotic DNA, the circular DNA, so of course in this case, what is going to happen? In this case, there is the formation of replication bubble. What will form here, my dear people? Replication bubble. So this replication bubble is nothing. It is having two replication forks, right? So why is it so? Because here the DNA replication is bidirectional. Here the DNA replication is bidirectional. It is taking place in both the sides. It's not like that. Firstly, this first strand will get copied, then the next strand. So it is bidirectional. So if you will look at this part here, this part here, this part here, this part here, you'll see that it appears like two Y-shaped replication fork. So here, this is first template strand. This is one template strand, my dear people, right? This template strand is having polarity 3 dash to 5 dash. This is another template strand. This template strand is having polarity 5 dash to 3 dash. This template strand is having polarity 5 dash to 3 dash. 5 dash to 3 dash. Now this is the Y-shaped replication fork. Now you are trying to open the DNA. It is helically bound. You are trying to open it. So of course, even if you will try it at home, anything which is helically coiled, when you try to open it, some supercoilings will form, right? It will get folded in such a way that you will not be able to open that structure. So in that case, we need the enzyme that can cut that supercoilings, that can remove that supercoiling, so that our helicase can open up the DNA, right, easily. That is also one of the point. Secondly, when you are trying to open up this DNA strands, right, they'll try to rejoin with each other, and this is not something that we want, right? This is not something that we want. So what do we need here? SSBP. What are these SSBP? They are single stranded binding proteins. What are they, people? They are single stranded binding proteins. Yes, my dear students, what are they? They are single-stranded binding proteins, right? They will not let it, do not let DNA reanneal, means to rejoin, right? And then one more thing, you have to remember one more category of enzyme that is known as topoisomerase. What's that? Topoisomerase. This is the one that removes supercoiling. Right, this is the one that removes supercoiling, right, bache? So here when we talk about topoisomerase, we have two classes, class 1, class 2. One will break one strand only, right, and then, remove it. It can make a cut in both the strand, then remove it. That is not the part of our concern. So topoisomerase is the one that will remove that supercoiling. And we talk about one more enzyme, that is DNA gyrase. It is DNA gyrase. It is not DNA gyrase. It is the DNA gyrase that we have. It is the DNA gyrase that we have. Are you getting my point? It is the DNA gyrase that we have, my dear people. And this one is it is a class 2 topoisomerase. So this is the one that will remove supercoilings in prokaryotes. It introduced the negative supercoilings. So it removes supercoiling in prokaryotes. ठीक? In prokaryotes. Okay. Now, the main replicating enzyme, that is the DNA polymerase. And here, I'll use the short form here, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Actually, my dear students, this is the main replicase, or I would say it is the main replicating enzyme. It is the main replicating enzyme. In prokaryotes, I'm talking about. In eukaryotes, we have different DNA polymerases. In eukaryotes, we have different polymerases, DNA polymerase alpha, beta, and all. Now, this DNA polymerase 1, on the name of the scientist, it is also known as Kornberg enzyme. See, beta, they all can polymerize. That is not the point. And when it comes to the polymerase activity, that's how we write 5' to 3' polymerase activity. Actually, we have to understand three things here in all these enzymes. If anybody is studying this topic for the first time, then it's my suggestion to watch my detailed lecture. You will have better idea of that. So 3' to 5' polymerase activity means it will move from, 5' to 3' polymerase activity means it will move from, it will synthesize the bases from 5' to 3'. Now, other than that, there is one 3' to 5' exonuclease activity. There is one 5' to 3' exonuclease activity. And this particular property, it is unique. It is unique to DNA polymerase 1. Right? This property is unique. Now, let me explain you this topic. All these three enzymes can synthesize the DNA. It's not like that they cannot. But this one is the main replicating enzyme, because we do not want the enzyme to be fast. First of all, there are two conditions. The enzyme has to be fast and it has to add the bases accurately. Otherwise, mutation can occur. I'm repeating my point again, people. I'm repeating this point again, right? The enzyme, right? I'm repeating my point again, that the enzyme has to be accurate, plus accuracy and the speed, both things are important. Let's say the enzyme is fast, but it is not adding the bases accurately, then it will be incorrect, right? Then it is not true, it is not good, it is not fine, it is not right. Are you getting my point? So it has to be fast, it has to be accurate. That's why we have the DNA polymerase 3. We talk about the concept of fidelity here, but let's not discuss that now. All the three enzymes. Now, when it comes to, let's suppose this is the enzyme which is synthesizing the strand in 5' to 3' direction. The enzyme is synthesizing the strand 5' to 3' direction. Now, let's suppose it is adding the bases ultimately. What is it doing? It is ultimately adding the bases. So, let's suppose, imagine you're organizing something, there is a line. Right, you are organizing something, there is a line, right? You have to cross check that the sequence is perfect or not. So let's suppose from A point to B point you are moving, right? You are arranging something. Now you have to cross check. You can simply move from B to A, right? You can simply move from B to A. What you people can do, you can simply move from B to A point, right? So that you can check, simple as that, right? Now, my point here is, right, no doubt enzymes, they will add the bases in 5' to 3' direction. So when they need to do the proofreading, right? What they need to do? Proofreading, right? So which activity of the enzyme? 3' to 5' exonuclease activity will be there. So that helps in the proofreading. Right, that helps in the proofreading. Now, we have one more enzyme, right? So I'm saying all these property, all the enzymes are having, like, when you talk about these two things, this and this, they all are having this property. But when it comes to 5 dash to 3 dash exonuclease activity, what is the meaning of nuclease? Matlab it will remove nucleic acid, nuclease that can digest nucleic acid. Exonuclease mane terminal, matlab it can remove the bases from ends. It can remove the bases from ends, right? It can remove the bases from the ends, right? From the ends. So 5 dash to 3 dash exonuclease activity means this enzyme will move from 5 dash to 3 prime direction. It will move from 5 prime to 3 prime direction, and it will remove the bases. What will it do? It will remove the bases from that particular end. Why it is used? It is used for primer removal, right? This activity is used for primer removal. Then it's 5 dash to 3 dash polymerase activity is used for gap filling. So actually, right? So this enzyme is very important for primer removal and gap filling. And when it comes to the enzyme which is having a very big role in the DNA repair, or they say all three helps in the proofreading. So mainly for the DNA repair, this DNA polymerase 2 will help. You can keep it in your mind. Okay. Chaliye. Now you know the main replicating enzyme, that is DNA polymerase 3. So what will it do? It needs to add the bases, right? What bases? You know that we need the deoxyribonucleoside monophosphates. This is something that we need to add in the newly synthesized DNA. Deoxyribonucleoside monophosphate. So one thing that you have to keep in your mind, this DNA replication is going on here in the cytoplasm. So this cytoplasm is having right? So what we need to add? We need to add DAMP, DCMP, DTMP or DGMP in our DNA strand. They are the monophosphates that are the part of our nucleotide chain. They are the monophosphates that are the part of our nucleotide chain, right? Matlab, whenever we talk about the DNA, like let's say you are saying adenine is forming 2 hydrogen bond with thymine, cytosine is forming 3 hydrogen bond. So actually it's a complete nucleotide, na? Right? And it is having single phosphate. So can I say that it is DCMP that will attach here? It is DAMP that will attach here? Yes, it's true. It's true. But basically here, right here, what will you get? Like let's suppose if it is prokaryote, it is a cytoplasm. So DATPs, DCTPs, DGTPs, DTTPs, they are present. Triphosphates are present. So they have the dual function. Matlab they are the triphosphates. They will act as substrate, right? They will act as substrate, plus they'll provide energy for polymerization. Matlab these are the triphosphates. And what do we need in our chain? We need the monophosphates. So it is the substrate, right? It is the substrate that we have to add in the, it is something that we have to add in the newly synthesized DNA. It is something that we need to add there in the newly synthesized DNA, right, bache? Newly synthesized DNA. So it is the triphosphate. So of course we have to add it. Let's suppose it is DMP. The two phosphates, they are the high energy bonds, right? So when basically we have to add DMP. So basically when these, right, triphosphates will get added, the 2 high energy phosphate bonds, they'll release the energy. That energy will be used for polymerization. Are you getting my point? That energy will ultimately be used for polymerization. Are you getting my point, people? Are you getting my point? That energy will be used for polymerization. Are you getting my point? I hope it is clear to everybody here, right? Okay, number 1. Now, that's your DNA. Okay, that's your DNA. Again, I'm repeating, this strand is having polarity 5 dash to 3 dash. This strand is having polarity 3 dash to 5 dash. This one is having 5 dash to 3 dash. Take synthesis will always be in 5 prime to 3 prime direction. It doesn't matter that whether it's DNA or it's RNA. Synthesis will always be there in 5 prime to 3 prime direction. Now, the main replicating enzyme is DNA polymerase 3, but it is having the drawback. It needs 3-OH to add the bases. It cannot start the DNA synthesis by itself. It cannot start it from the starting. I'll put the first break, then I'll put the second. No, it cannot. It's not its cup of tea. It needs the 3-OH group. Whenever your DNA polymerase 3 will see the 3-OH group, it will be able to add the further bases. It cannot start the initiation by its own. It needs the 3-OH, then it will be able to add the bases further. That is my point. Okay, that is my point. So for that, right, we need one more enzyme that is known as primase. It is because it uses the DNA template. So it is the DNA dependent RNA polymerase. It is also known as DNA dependent RNA polymerase. ठीक, so it will form primer. RNA primer, right? A short nucleotide sequence, like 15 to 20 nucleotide sequence ka RNA primer. So as I said, synthesis will always be in 5' to 3' direction like this. So 3' end means free OH group. So your DNA polymerase 3 will be able to add the bases further, right? Because it will see this primer. Primer is having 3-OH group. So it will be able to synthesize add the bases further, right? That's the point. Now, the second thing is, when you talk about DNA polymerase 3, it's not like that it will come back for this strand. It has to do the synthesis of both the strands simultaneously. Then what is going to happen here, my dear people? So DNA polymerase 3, dekho, this is the direction of replication fork. Right? Helicase is moving like that. It is opening the DNA helix like that, right? Your DNA polymerase 3 is also moving like that. DNA polymerase 3, number 1, it needs 3-OH to add the bases, plus it has to synthesize the two strands simultaneously, right? But in opposite direction, right? It is not capable of doing it. Okay, fine, if I have to move in this direction, I can synthesize the strand in this direction, right? But in this direction also it has to synthesize. So here, the template strand is 5' to 3'. So opposite to it, right? The new strand that will synthesize, right, opposite to it, the new strand, beta, that will synthesize. First of all, this strand is opposite to the direction of replication fork, number 1. This strand is opposite to the direction of replication fork, right? Keep it in your mind. This strand is opposite to the direction of replication fork. Why? Because your replication fork is moving in this direction. We have to synthesize it in opposite direction. So ye dekho, 5' to 3', 5' to 3', 5' to 3'. Now imagine, for DNA polymerase 3, it is difficult. It is moving in this direction, but it has to synthesize the strand in this direction, and it has to synthesize one more strand here, and that too in opposite direction. This strand is going in this direction. This strand will get synthesized in this direction. So this strand is not at all continuous. It's a discontinuous strand. What kind of strand it is? It's a discontinuous strand. Right. It is also known as lagging strand. And the fragments that are getting synthesized here, they are on the name of the scientist. So we call them as Okazaki fragments, because polymerase 3 cannot synthesize the strand in opposite direction, and that too, right? That too continuously. So that is how it will be. Take. So it is the discontinuous strand, lagging strand it is. So again and again here, we'll be needing the primer. Primer is required here. Here it will be continuous strand. So in the paper, this kind of question can come, right? The question can be like this, right? That which template strand, complementary to which template strand, leading strand will form. It is the continuous strand, also known as leading strand. So leading strand, right? Leading strand, it forms complementary to, right? It forms complementary to template strand, it forms complementary to template strand having polarity 3 dash to 5 dash. That can be asked as a question. That can be asked as a question. Right? Right? That can be asked as a question. So, leading strand, it forms complementary to the template strand having polarity 3 dash to 5 dash. Right? This is what you have to remember. Now, let's suppose this synthesis will occur. Now, in between RNA primer is also there. So, we need to remove that RNA primer. Right? So, who's going to do that? Who's going to do that? Your DNA polymerase 1. It's 5 prime to 3 prime exonuclease activity. It will help in primer removal. So when primer will be removed, we need to fill these gaps. So DNA polymerase 1 will add the deoxyribonucleosides monophosphate there. Right? DNA polymerase will do it. Now after that beta, DNA ligase will help in binding the sugar phosphate backbone. Right? So it is the DNA ligase. I hope you remember what DNA ligase is. It is also known as molecular glue, my dear people. Right? It is the molecular glue, my dear people. And this particular enzyme, it binds sugar phosphate backbone. Right? This molecular enzyme, what will this enzyme will do? It joins that sugar phosphate backbone. What will it do? It will join that sugar phosphate backbone. What will it do? It will join that sugar phosphate backbone. That's what you need to keep in your mind. Take. So it is all about the DNA replication. Solve the questions on the basis of that. Right. This is your lagging strand. This one will synthesize first, second, and third. Discontinuous synthesis. Template strand is having polarity 5 dash to 3 dash. Continuous synthesis. Template strand is having polarity 3 dash to 5 dash. Take the role of enzymes should be clear. Now look at this in diagram. Look at this enzyme. Please have a look. Everybody, please have a look. Please have a look. Done? Sure? So, DNA polymerase 3. This is the leading strand. This one is lagging. It has to do it again and again. Again and again, primase will form primer. Again and again, DNA polymerase has to synthesize the strand. So, DNA polymerase 1 is for what? It is for the primer removal, right? It is for the primer removal and then gap filling. So it is the 5 dash to 3 dash exonuclease activity that will help there. That will help there. So that is what you need to keep in your mind, people. So this is all about the topics today. Here you have the flow chart where, right? Here you have the table where all the enzymes and their functions are mentioned. Okay, DNA gyrase, primase, everything. Right. Other than that, do revise the Mendelian disorders. You can just simply put the disorders by Ambika ma'am. So there will be a session, right, in which I have explained all the disorders. Okay, so you can just check it. This is all about the today's session. Rest everything we will discuss in the marathon. So you know that tomorrow, right? It's still tomorrow. Tomorrow at 8 p.m. right? A session will be there, right? Where you'll get to know about the details of all that marathons. So stay tuned to that beta. And if still you are struggling with the revision, you don't know which chapters you need to prioritize, you people can watch this particular video. Okay, 45 chapters that you need to study, that you need to master for 650 marks. And here beta, in this, right? In this description box, you have the link, right? You have the link for the test series as well. Okay, NCERT Crux test series. So see you all right? See you all, and do recommend this channel to your juniors. Tell them there is an amazing channel where teachers are taking marathons, they are taking short session, long sessions, so that students can understand the topic. Okay, so we are here to help you guys. We will be there for all of you, so do not forget about this. And tomorrow, okay, yes, tomorrow there is a session, right? Tomorrow there is a session in which what will be done, my dear people, right? The basics of biology for 10th moving to the 11th student, right? There will be the basics of biology. So if you like the session, do let me know in the chat section, right? Do mention it in the comment section that you liked it. So take care. Thank you so much, people.